Good morning, good afternoon, and good evening to all of you, depending on where you are. Thank you for joining us today. My name is Neringa, and I will be your host for today's webinar entitled Accurate Vector Copy Number Determination in Gene-Modified Cellular Products via TLA and ddPCR. And at the end of a session, there will be a Q&A panel available where you can write down any questions, and we will go through them at the end of the webinar. However, if time will run out, we will make sure to email you the answers afterwards. So let's move to the first slide. So I believe we are all very well aware that cell and gene therapy is a revolutionary and rapidly growing field. So as you can see on the top left image, that considerable CGT activity is happening in the North American region. And these data are taken from Alliance for Regenerative Medicine last year. And what you also notice on the bottom right graph, that lentivirus is clearly the most popular vehicle for gene therapeutic delivery. And in fact, retroviral and lentiviral vectors continue to play a critical role in the development of gene-modified cell therapies, and especially T-cell therapies. And talking about the T-cell therapies, so arguably the most popular example is CAR T therapy, where the process involves collecting T-cells from the patients. Once you have those cells collected, you re-engineer them in the lab to produce proteins on their surface called CARs. Once you re-engineer the cells, you want to grow millions of CAR T cells and reinfuse these engineered cells into the patient. However, because vectors which are used to engineer T cells can permanently integrate into the host cell's DNA, comprehensive QC of their genetic outcomes becomes essential to guarantee safety, efficacy, and consistency of those cells. Therefore, vector copy number per cell represents a critical validation parameter for product characterization during ATMP manufacturing. And studies have shown that the risk of oncogenesis increases with higher copy number per cell. So due to this reason, the FDA recommends limiting vector copy number, which is up to 5 copies. And therefore, droplet digital PCR or ddPCR has proven to be an accurate method to precisely quantify the number of vector copies integrated per cell. So before we will move to the case study, for those who might be not familiar with us yet, I would like to give you a brief introduction. So we are a Dutch company founded in 2012. Actually, we are a spin-off from the Hubrecht Institute, and we are located in Utrecht, the Netherlands. And we focus on providing genomic analysis services to biotech, pharmaceutical companies, and academics based on our technology known as targeted locus amplification, or TLA. And TLA, coupled with next generation sequencing, stands as a QC tool for the comprehensive genetic characterization of transgenic cell lines, animal models, as well as for gene and cell therapy products. And since last year, we have joined Solvia's group, and Solvia's has a comprehensive analytical testing portfolio and capabilities to support all stages of drug discovery, development, and commercial release. And in this slide, I would like to get back to the genetic characteristics. So the most relevant genetic parameters you can see summarized on the left side of this slide. And on top of the slide, you can see a range of different technologies which are used for the characterizations. And of course, all of these technologies have their advantages and limitations, and it really depends on the research questions you have to answer it. So basically, each method allows you insight in only one or few parameters of interest, but to get a comprehensive picture, you will need to use combination of several methods. So a key distinction of our TLA-based approach is our ability to check both transgene integration site and the integrity of integrated sequence in a single experiment. But however, as you might know, TLA only provides an estimation for the copy number. And since the FDA puts a strong recommendation to determine the precise vector copy number, we have worked on a solution to determine it. So by coupling TLA-based data with ddPCR results, we are able to get the precise vector copy number. And in the following slides, we will use the NIST project as a case study. So NIST stands for National Institute of Standards and Technology. And since 2021, we have had the pleasure to join the NIST Genome Editing Consortium. This consortium brings together 40 leading experts across industry and academia. And this involvement reflects a shared commitment to improve the quality of genetic research. So one of the NIST aims is to evaluate and qualify robust analytical methods to assess genome editing outcomes, and more specifically to identify common measurements and standards to improve confidence in characterization output. So today we will present to you a NIST Interlab study, which goal was to assess lentiviral copy number and integration sites in 5 transgenic human cell lines. So for this study, we received 5 URCAT cell line samples, and these samples had a different number of lentiviral vector copy numbers integrated. These cells, they're transduced with a lentivirus expressing GFP, and the number of integrated copies varied from 1 to 4 copies in the cell lines, and we also received a control sample. I would like to highlight that we received blinded samples, so we didn't know how many copies were present in each test sample. And as mentioned, the goal of the study was to determine precise vector copy number and integration sites. So in order to answer those research questions, we coupled TLA-based analysis with ddPCR data. And we started with TLA analysis to determine specific location of integration sites, number of integration sites, and estimation of vector copy numbers. After the TLA analysis, we performed ddPCR assay, which was developed based on the TLA data. And using the ddPCR analysis method, we were able to validate integration junctions and precisely determine the vector copy number. And what I would also like to emphasize in this slide, that we could also design ddPCR assay without TLA data, and it is represented in example 1. So in this case, ddPCR assay would be designed on transgene or common vector elements. So those assays design is indicated in pink, and on the reference gene, an assay design spot is indicated in blue. And in most of the cases, actually, this assay design works perfectly fine if your reference gene is deployed and you're determined 2 vector copy numbers as expected. But however, if selected control shows unexpected results, using the TLA data for the ddPCR assay development gives an advantage to design a more precise assay. So as you can see in example 2, using the TLA results, we designed ddPCR assay not only on transgene or vector common elements and reference gene, but also on the vector-vector or vector genome junctions as an additional control. But before moving to the TLA data, I would like to explain the TLA method itself. So how does the TLA work? So basically, once we receive the samples, we start by cross-linking viable cells using formaldehyde as a cross-linking agent. And in case if we work with virus-producing cells, we ask to provide us with cross-link sample. After cross-linking step, follows the steps of restriction using extraction enzyme and ligation to create DNA circles. And essentially, during previously described steps, we reshuffle the DNA to create circles that contain fragments of digested DNA. And TLA relies on the proximity ligation principle, so meaning that the sequences that were originally close from each other will have a higher chance of ending up in the same circle. But however, it is still very much stochastic process, so wherefore generated TLA circles don't have a predefined composition. So once we have DNA circles, next comes the enrichment step. So here our experts will design inverse primers that will be specific to a specific sequence of your locus, for example, your transgene. And as such, TLA-based approach will be able to selectively amplify only the circles of interest in order to generate large coverage across region of interest. So the assay readout looks like depicted in this image. So the generated sequencing coverage looks like a bell-shaped curve, and coverage will be highest next to the primer binding site and decreases the further away the sequences are from the design primers. So with this method, we are able to identify the integration sites, structural changes surrounding the integration sites, and single nucleotide variants. TLA method has been published in Nature Biotech. So if you would like to better understand how the TLA approach works, I would highly recommend you to look into it. So as mentioned during the NIST study, we have tested 5 blinded samples, and different samples were named VCN 0, VCN 1, 2, 3, and 4. And in this slide, we would like to show you the TLA data. So in the graph A, we have an information on genome-wide coverage plots. And this is the plot of the entire genome with chromosomes listed on y-axis and the chromosome position listed in the x-axis. And although you can see the coverage everywhere, I believe you can clearly see an enrichment at specific locations. So there are also in circles indicated the integration sites. So basically here you can see where your vector has been integrated, and whatever it is a single integration or multiple integration. And I would also like to emphasize that all these samples were tested independently, and we only combined the data of all these samples for representation purposes. So actually the data was as expected. We haven't detected any integrations in the control cell line, VCN 0, and we detected only 1 integration site in VCN 1 sample, 2 integration sites in VCN 2 samples, 3 in VCN 3 sample, and 4 in VCN 4 tested sample. And only once we have determined specific integration sites of the samples using the TLA, we have also compared our data with previously published NGS data, and this comparison is depicted in the table B. So basically what we can see from this table is that with TLA method, we found the same integration sites as published in 2021, except of a 1 integration site in the VCN4 cell line. So this specific integration on the vector appeared in the highly repetitive region, wherefore we are quite confident that we were able to detect the correct integration location, since TLA is a very precise method. And there is a high chance that due to repetitive region, published data was mapped to the wrong region. As mentioned before, using the TLA method, we are able not only to identify integration sites, but also to determine any single nucleotide variations or genomic rearrangements. So a single small sequence variant was identified in the integrated vector sequence of cell line VCN1, VCN2, and VCN4. And based on the mutation frequency, which was 20% for VCN4 sample, 50% for VCN2 sample, and 100% for VCN1 sample, we concluded that these variants are only present in one of the integrated vector copies. Regarding the genomic rearrangement analysis at the integration site, a 648 kb deletion was detected near the integration site on chromosome 14 in cell line VCN3, and no genomic rearrangements were detected at the other integration sites. Here I want to show you the ddPCR assay design process. So we have designed ddPCR assays on one of the listed reference genes, on the breakpoint sequences, and on the vector-specific elements. And assays were designed in a way that probe would bind to vector sequence or breakpoint sequences as indicated in this image. For the ddPCR analysis, we use QX200 ddPCR system from the Bio-Rad. And Bio-Rad ddPCR system is known as a gold standard method. So it has a wide dynamic range as well as multiplexing capability. And here we have steps in a typical ddPCR workflow. So basically, first is the preparation of ddPCR reaction mix, in which your template, primers, probe, and PCR mix is combined together. Next is the droplet generation step. So the ddPCR mix is combined with the oil to generate droplets in the ddPCR droplet generator. First step is PCR amplification. So during this step, PCR reaction is performed in every generated droplet. And finally, you read your samples in ddPCR analyzer and analyze the results. And what it is important here is to have a clear separation of your droplets. So negative droplets will end up in the bottom left corner. Vic or FAM positive droplets will be placed on top left or bottom right. And double positive droplets will end up on top right. In the graph here, you can see that expected copy numbers were detected in all cell lines using the ddPCR method. So same as with TLA results, we haven't detected any vector copy numbers in VCN 0 sample, and 1, 2, 3, and 4 vector copy numbers in tested samples. So I would like to highlight that reference copy number must be known for the accurate quantification. And both genomic genes and breakpoint sequences may serve as a reference. So take-home messages. So for copy number assessment, the results generated with ddPCR aligned with our TLA-based data. Breakpoint sequences identified by TLA, they're validated with ddPCR assays. For accurate copy number assessment, we recommend using the breakpoint sequences as a reference instead of the reference genes. And although sequence variants were detected in VCN1, VCN2, and VCN4 cell lines, these mutations did not affect copy number assessment. So by coupling TLA and ddPCR results in this study, we have performed the most comprehensive analysis of tested samples compared to the other participants. So regarding our ddPCR services, we have already done several projects, including precise copy number determination and stability testing, also endogenous gene copy number determination, presence of residual plasmid, as well as translocations at the predicted cut sites. And as mentioned in the previous slides, for the transgene copy number determination, based on the TLA results, we will design ddPCR assays on the transgene or another vector element to measure the copy number and on the reference gene. And in addition, and if it is possible, we will design a ddPCR assays on the integration junction of a transgene as an additional control to validate the integration sites. So finally, here is an overview slide touching upon our main expertise, in-house capabilities, and experience at Surgentis. So at Surgentis, we have a dedicated team of PhD level scientists. We are also ISO accredited, which demonstrates that we operate our services within a robustly reliable, compliant framework. We have a pleasure to support leading biotech and pharma globally in over 40 different countries. And with over 10 years of experience, we had the pleasure to make world-class partnerships, and the most recent one being the release of a joint white paper with Horizon. And our unique genetic insights continue to heavily endorse in peer-reviewed scientific publications. So with this slide, I would like to thank you all for your time, and we hope that this webinar has been useful and informative and has given you a better understanding of how we help our customers with accurate vector copy number determination at Surgentis. So with that, I would like to conclude and thank you all again for joining us today.